In this module, we'll be going over Broad best practices for keeping up your GA and how to solve some common problems when your flow cell is actually loaded. First, we'd like to demonstrate a Broad best practice of GA maintenance. We do a sodium hydroxide wash before every run to ensure that lines are clear and make sure we don't lose a run due to a clog. 1N sodium hydroxide kits are made for every instrument and are reusable. These include bottles for every active port on the sequencer, including those on the GA and the paired end module. The kits are made from diluted 5N sodium hydroxide and are then aliquoted into the appropriate sized tube for washing. Since we reuse our kits at the Broad, we label each tube with the appropriate port number to prevent any contamination. Illumina recommends washing once a month, but due to our high turnaround, we perform a sodium hydroxide wash before every run. As part of the sodium hydroxide wash protocol, we perform a water wash before and after the sodium hydroxide to flush out the system properly. When your old reagents are taken off, load the sequencer with a water wash first. When loading either the water or sodium hydroxide at its appropriate time, you want to make sure the volume of the liquid is high enough to cover the entire sipper to clean it. The series of events is a lower volume wash, prewash.xml with water, a lower volume wash, same recipe as before, prewash.xml with sodium hydroxide, and a high volume wash, postwash.xml with water. When the first water wash is complete, then the tubes of sodium hydroxide should be switched on using gloves. At the end of the last high volume water wash, the maintenance sodium hydroxide wash is complete. The sequencer is ready to be loaded with reagents for a new run, or it may be left with water in the lines until needed. One of the common adjustments we make to the GA is footprint adjustment. The beam shaper assembly is responsible for the linear representation of the laser footprint against the prism. There are a few adjustments you can make which might be necessary depending on several factors, like when your last PM was done, variances in flow cell construction, how the flow cell is seated on the stage, or if you accidentally bumped the beam shaper assembly during flow cell loading. There are two key adjustment points that you can work with to make corrections to the image. These are the translational stage adjustments, which adjust the image in the X and Y directions. Don't change the other screws because these should only be adjusted by a service engineer. These other screws will actually change the image offset instead of just adjusting the linear direction of the image. Turning the top screw clockwise adjusts the image in a left to right direction. Turning the bottom translational screw clockwise adjusts the image from top to bottom. We'll demonstrate making some subtle changes to each of these screws that will throw the image out of alignment. We've positioned the flow cell at lane 4, column 1, row 25. We'll take a picture and you can see the image is black on the bottom and left edges. If, after loading your flow cell, you see something like this image, the first step should be to remove the flow cell from the instrument and reload it. This makes sure that the flow cell and prism are seated properly and that proper oiling technique was used. If, after reloading the flow cell, the image still looks like this, you should check different lanes of the flow cell to ensure that you are getting consistent representation of the misalignment. Here you can see, as we move to different lanes, the misalignment seems fairly consistent. The next step is to adjust the footprint. Go back to lane 4, column 1, row 25, take a picture and note the visual position of the footprint. Knowing that the top translational stage is responsible for moving the image left and right, we'll adjust the top screw counterclockwise since we need to move the image to the left. Make small adjustments at first until you get a feel for how much movement of the image is associated with a given amount of rotation of the screw. In this case, about half a revolution of the thumb screw was enough to get the image to look like this. We now adjust the bottom thumb screw about a half revolution clockwise to bring the image down. We want to ensure that the image is good in both column 1 and in column 2, and that we're not cutting off too much of the image in either column. Because we're demonstrating with a beaded test flow cell, the right side of column 2 is dark, but this won't be the case on a real flow cell. We'll check lane 2, column 2, and check that image as well as check lane 4 and lane 6, and you'll see that we are fully compensating for footprint adjustment. We can fine-tune the image and turn the bottom screw a quarter turn clockwise, which is responsible for adjusting the image in the Y direction or up and down. One caution when you load a flow cell and you see that the footprint seems to be exponentially growing either from lanes 1 to lane 8 or from lane 8 to lane 1, an issue called footprint walking, then you need a more detailed adjustment. If this is the case, then you should make a service call to your local Illumina service person to make these adjustments.